Okay, welcome everyone. Maybe someone can close the door, then we don't hear the kitchen. So I'd like to give a short play in German. Kürzlich wurden die geheimen Sitzungsprotokolle vom Modellvorhaben Genomsequenzierung veröffentlicht. Und ich möchte Ihnen ein paar Auszüge daraus vorlesen. Jens Spahn begann damit, dass er das Modellvorhaben zu einer geradezu nie wiederkehrenden Gelegenheit erklärte, die zu verwirklichen man für das Wichtigste und Größte halte. Wir müssen und werden ganz große Idee verwirklichen. Wir haben die Gelegenheit und dürfen uns ihr nicht entziehen. Denken Sie an was Bestimmtes? Fragte ein Staatssekretär. Nein, er dachte nicht wirklich an was Bestimmtes. Wie hätte er das auch tun können? Niemand, der vom Größten und Wichtigsten der Welt spricht, meint, dass es das wirklich gäbe. Sie haben wahrscheinlich mittlerweile erkannt, dass es sich nicht um einen Protokollauszug handelt, sondern um Textpassagen aus dem Mann ohne Eigenschaften von Robert Musil, in dem die große vaterländische Aktion beschrieben wird, bei der jeder dabei sein möchte, ähnlich also wie im Modellvorhaben. Auch zur Umsetzung finden sich in diesem Buch wertvolle Hinweise. Größtenteils entsteht Geschichte aber nicht von einem Zentrum her, sondern von der Peripherie, aus kleinen Ursachen. Wahrscheinlich gehört gar nicht so viel dazu, wie man glaubt. Deshalb freue ich mich, dass ich Sie hier in Bonn, dem alten politischen Zentrum, mittlerweile aber der Peripherie zur AGD 2023 begrüßen darf und hoffe auf anregende Diskussionen, um die große vaterländische Aktion zu verwirklichen. Jetzt noch ein paar Housekeeping Rules. So, you all have seen that posters are online, and there is there prizes for the best three, and please vote until tonight so that we can have the winners give small talks tomorrow. Then all the talks from the conference will be available until November 14th in the online platform. And tonight we don't have a concert, but a games night. So you will see 4 suggestions on the back of the program. So if you are interested in a particular one of them, try to identify the host of the game in the breaks. And I also wanted to point out an interview study that Helen is conducting. So everyone familiar with Face2Gene, the app, as a clinician that wants to improve that app for the next iteration, please get in contact with her, because there you can provide feedback, which we will use for the next interface. Okay, now I would like to hand over to Joachim, who is the chair for our first session, which is about the highlights of the next generation competence centers.
Thanks a lot, and it will not be as philosophically and as exciting. I like that very much. Very good start. Thanks a lot. I think I have to read more books now again and not work only. So it's my pleasure to introduce you some highlights of what is still called the DFG-funded NGS competence centers. We're running in year 4. This is a project, and we're still in place until 2024 at the end. That's decided just 2 days ago that we got prolongation. And probably beyond that now into 2025. Unfortunately, as it is with DFG, very often it's called cost neutral extension. So we have to do the work, but without money. I'm not complaining, but I'm just saying that this is a structure that has some value developed over the last 4 to 5 years. And I can also tell you that there is now movements in the different ministries that might or might not support extension of such structures, and it goes along, Peter, the lines that it's not centralized. It's they want to basically foster genomics across the country in a peripheral or decentralized fashion. Yeah, so we're working on that along the lines, and this is just coincidence, but I would like to mention that. And without further ado, we have 4 presentations from the different centers, and we start this time with Dresden, so the most east center of those NGS competence centers. And this is the only one online. This is Rebekka. Fabian Rost will present us. They did not come here today, but we have hybrid, so they will present there as an example from the center, characterizing the immune architecture of melanoma metastases, a spatial multi-omics approach. Fabian and Rebekka, please, the stage is yours.
So, can you hear me without problems?
Yes.
Perfect. So, okay, so it's a pleasure for us to present to you some very new data we generated in the last months, and we want to characterize the immune architecture of melanoma brain metastases in a multi-omics approach. And I think most of you know that cancer cells not just live by their own as an aggregate in the body. They are in close contact to a lot of different other cells, and that's very complex microenvironment that consists, of course, of the tumor cells, but they need vessels which supply oxygen and nutritions. We have a lot of cells that form this, they help to structure the whole thing. We have cancer-associated fibroblasts. We have the extracellular matrices that make everything possible. We have immune cells, a lot of immune cells in there, where we have the macrophages, we have T cells and dendritic cells. And in the brain, we also have, when we are with the phagocytes, we have the microglia, we have the nerve cells. So a lot of cells that can promote tumor growth, but they can also help to kill tumor cells and promote the survival of the patient. And we here now have one case in the clinic, which is a long survivor of a melanoma patient with a brain metastasis. And we got tissue of this brain metastasis and were able to analyze this. So we got formalin-fixed paraffin-embedded tissue. And after surgery, the brain looks like that, or the tumor looks like that. And the transcriptomics we performed on the Xenium from 10x platform, which is very new, and the proteomics we performed on Maxima imaging platform from Milteney. But I now want to first introduce to you the Xenium platform, which is very targeted. So we have padlock probes where we have sequences which are very specific for the transcript or for the we want to look at. And if the binding is stable, the probe is ligated. We also have here the gene-specific codes there, which then are amplified in a rolling circle. And the whole barcodes can then be visualized with fluorophore coupled nucleotides and analyzed in the xenium. And what we get at the end, I think Fabian will give you some more details. We have DAPI staining in addition, where we have the nuclear segmentation, and we also have a cell segmentation. So what we really get here is transcriptome data on a single cell level, and we have the spatial distribution. So we know who is the neighbor of the cell we are interested in, and that gives us a quite new step in how the architecture of a tumor can look like. So the Maxima platform looks for proteins and with cyclic staining, and we stain with antibodies. Unfortunately, we were not able to analyze the whole tissue, what was possible in the xenium. We went for regions of interest, and we stained in cycles. So we have directly fluorophore coupled antibodies, which we have 3 in one cycle plus DAPI. We image everything, and then the fluorophore signal of the antibody is bleached so that we can start with new fluorophore coupled antibodies with specific signals in a new round. And that everything is automatically stained in the system and provides quite robust data. And what we now want to do with that, I just give you a short impression of that. We can now look for the same areas in the transcriptome and in the proteome. So we have here, unfortunately not on the same section, but we have serial sections. And I now did the registration, or put them somehow together with the help of this vessel, so which we stained in both systems with the help of CD, or visualized with the help of CD34. And as you can see, there's a lot of more cells in there. So we have a lot of T cells infiltrating the tumor tissue, which we see with CD3 stained on the proteome level, and the transcripts of CD3E and D in the xenium. And in both systems, we get the same regions infiltrated with T cells, and we also see in both regions the aggregate of B cells, which are visualized with CD20 on the xenium with the gene with the MS4A1. And we have see the proliferating cells with Ki67, which are in this case the T cells, which is also true for the xenium data. So what we can see that we can that I can trust my antibodies. So that is the simple and the first thing I was happy about. But of course, we can now also see how reliable our transcriptome data. Many studies look at bulk transcriptomic data, and you don't know it's this high transcript level due to what does it mean in a protein? Let me say like that. Do high transcript levels also mean high infiltration, for example, of T cells when we have a lot of CD3E and D, for example? And of course, why do we do this? Because we get some information from the xenium and some from the Maxima and vice versa, which are in addition. So with the xenium, I forgot to mention that we have here a commercially available kit for 377 genes. So it's not the whole transcriptome. And for example, something like a senescence marker, the CD57, we can stain with the Maxima, which we don't find in the xenium, or an activating receptor like CD134, which we also find in the Maxima staining. But nevertheless, the CD8 cells, which are the cytotoxic T cells, which are very important in the tumor, you can nicely see them here. But you can also find cells which have inhibitory receptors like LAG3 here, for example, but also we find them back in the xenium. So, and what else can we do? we can deeply characterize the T cells with the transcriptome data and maybe find them back in the proteome. But of course, a lot of stuff is just visible on the transcriptome level, like targets like PD-1 and CTLA-4. We have potential new targets like TIM3 and 4-1BB, which we here can see nicely on the transcriptome data level, which I was not able to stain in this tissue, unfortunately. So it gives us a lot of information. And what I like to play around with is to see which molecules are expressed by which cells. So I'm a little bit more from the dendritic cell fraction. So dendritic cells are cells which nicely activate T cells, in the best case, which are specific for tumors. And in former times, when you just performed single immunohistochemistry, you stained with antibodies against CD38 or LAMP3, which characterize activated dendritic cells. And now we can put this together and see that these molecules, at least at the transcriptome level, don't mark the same cells in every case. So here are cells which have both molecules, and there are other more that just have CD83, and some that just have LAMP3. And when we have now a closer look, what cells might this be? So beside macrophages, there might be also B cells that express a CD83. And with that, you can step by step characterize your infiltrate and get a lot of information out of that. But how we start to do that, Fabian will tell you in a minute. I just like to show you some more pictures just from the Maxima platform. So where we have here a characterization of the microenvironment, where we have in white the tumor cells. We also have the vessels in there, which are stained with the CD34. There's some muscles, or alpha SMA positive cells around. We can characterize the tumor with different molecules, like here with vimentin or galactine-3. And of course, because I'm mainly interested in that, we can look at the immune cells. And here we have just a staining of the CD3 cells, which we can divide in the cytotoxic CD8 positive T cells and the T helper cells, which are CD4 positive. And you can nicely see that they are all around, and that we have proliferating T cells, and these are both fractions. So we have CD4 positive T cells as well as proliferating CD8 positive T cells. So a quite good hint what's going on in the tissue. We can also see at the resident T cells, which are characterized by the 103, and we see regulatory T cells. So there are also some immunosuppressive cells in there which express also FOXP3, but we also have T helper cells type 1, which do express TBIT, transcription factor, and they more promote an anti-tumor immune response. And on the other hand, we have cells of the innate immune system. There are macrophages. Here we stained for with the general marker CD68, which you see here in red. So we have a high infiltration, and we have CD163, which is more for the non-so-inflammatory macrophages. And in addition, we have just a few neutrophils in there, so just these two cells, but I can characterize them with 3 markers. That's why they are just white. So we have the CD66B, we have the CD15 and the arginase, which all look for neutrophils. And in between these macrophages, we also find, again, cells of the adaptive immune cells, which are the B cells. And there we have here in green, in general, the CD20 positive B cells, and we have plasma cells, which are CD138 and CD38 positive. So we get now an impression. So all these cells are stained on the same tissue, and we are now analyzing the data more in detail. And with that, I can draw a small conclusion until now. So I think a reason why this patient is so long-lived is because we have a massive infiltration of immune cells, which is quite rare. And these immune cells, like the Ki67 staining shows, are well activated, especially the T cells. And with that, I'd like to thank you for your attention and hand over to Fabian.
Hi everyone. Thank you, Rebekka. Thanks for having me here. So I'm sharing the talk with Rebekka as we're working on that closely together. I'll share my screen now. Can you see my slides? I think they're there. All right. I'm working at the Dresden Concept Genome Center, and I'm in the bioinformatics team there. And basically, I'm now like after Rebekka introduced to you the kind of situation we have with the patient. I wanted to talk about how we now try to like analyze this complex data in a systematic way. And I think for us the exciting thing is that this is like the spatial sequencing aspect, so that we now know information about where things happen in space. That's super interesting to us. Okay, Rebekka introduced to you the basic idea of this Xenium device by 10x. I just wanted to remind you what kind of data we get out of that, and we get essentially two things, which is the transcript data and the DARPy images. What do I mean by that? So if we look at the transcript data, here I try to illustrate the kind of amount of data that we have. So in the I think I should turn on. So here you see the whole tissue that we have, and I'm not sure whether you can see there's a little red dot down here. That's where we zoomed in, and this scale bar is like 50 microns, this white scale bar. And what I visualize here is like every dot is a single transcript. It's a single mRNA molecule floating around in the cells. And the different colors mean the different kind of genes to which these transcripts belong. So in this panel, we have 377 genes. And in the whole, like for the whole tissue, which is like 1 by 2 millimeters, what we see here, we have about 100 million transcripts floating around. And of course, that's way too much to look at it. So like to filter things out a little bit, let's concentrate on a few transcripts. And here I look at some that are relevant for T cells. So CD3 as a T cell marker, CD4 and CD8 for the different kinds of T cells. And we can see that there is some structure in this transcript. And so, yeah, for each of those molecules, we know where it is in space. That's one of the outputs that we get. And on the other hand, we get DARPy images out, so they mark the cell nucleus, and they give us an idea of where the cells are. And now we can do some we are a sequencing center, but now here we actually need some image analysis expertise to get things together, and that to me is super exciting. So what we need to do here first is to do some how we approach that is to do some nuclear segmentation. So we segment the nuclei to detect the single cells, and in the whole tissue that we looked at, we have about 1 million cells here by this. And then based on this nuclear segmentation, we do a cell segmentation. In this case, cell segmentation we just simply do by expanding the nucleus up to where we reach the next cell. And then when we put that together now with the transcriptome data, here again, just show the T cell markers, we can see now which transcripts actually belong to which cell. And for instance, here we find a cell that is positive for CD3 and CD8, so that's most likely a CD8 T cell. And now to systematically analyze these data, we take this segmentation together with the transcripts and put the whole thing into what we call a cell by gene count matrix. So here every row is a single cell that we segmented, and every column is a gene. And then for every cell, we just count the number of transcripts that we found. And so for this first cell, we, for instance, find 6 transcripts of the ACTG2 gene, and so on. And so we get this huge count matrix for 377 genes for 1 million cells. And on average, we find 100 transcripts in each single cell. Now, what I did here by condensing everything in such a count matrix is actually to throw the spatial information away for now. And we can now make use of well-established single-cell workflows. So I just here take a figure from a best practice single-cell workflow, where usually what we do is, right, where we come from non-spatial, we just sequence single cells and we come to a count matrix. That's what we now got from the spatial data as well. And then we do typical steps. I just quickly go over them without explaining much. Quality control, normalization, we select some highly variable features. And then at some point we arrive at a typical visualization of single cell data. And I'm not sure whether all of you are familiar with these kind of visualizations, but this is a first visualization of our data. And this visualization is called a UMAP. And in this UMAP, every dot, and there's now 1 million dots approximately on this UMAP, so you can't actually really see them, but every dot corresponds to a cell. And whenever 2 dots are next to each other, this means that they are transcriptomically similar. And why do we visualize our data like that? We want to somehow visualize, so these data are huge and complex, and we somehow want to see the structure of these data. And when I first did that, I was a bit disappointed, because basically what I was expecting is, as Rebekka already showed you, we can clearly see different kinds of cells. We can see T cells, different kinds of T cells. We can see B cells. We can see the dendritic cells. So I was hoping in this kind of visualization to actually then clearly see a separation of different cell types, and took us some, and we didn't see that. I was a bit disappointed, and it took us some time to actually get used to actually understand what we need to do. And I quickly summarize, so that has been quite some work with the lessons we have learned. So one thing is that one key thing was that we only use transcripts in the nucleus. So I showed you the cell segmentation before. This cell segmentation doesn't have a membrane staining, but only is based on the nucleus. So things can go wrong there. By only using transcripts from the nucleus, we actually lose 50% of our transcriptome data. So because only 50% of the transcripts are actually located inside the nucleus. But that was necessary. We also needed to be way more stringent on our cell quality control. What do I mean by that? Some cells, just by chance, we don't, or some cells do not have many transcripts. So we asked to have at least 50 molecules in a cell, and that seems to be what we needed given this panel to actually characterize that. That way, we actually lose 70% of the cells. So we actually lose quite some data when doing that, but what we definitely gain is a much clearer cell state structure. So going back to this visualization I showed before, that's the picture before, after learning these lessons. That's how things now look like. And here, this is what I tried to explain. Here we now can see clearly different regions of cells that group together because their transcriptome is similar. And so from that point, we wanted to go on further to now characterize what these cells actually are, and we are in the process of doing that. Usually what you do then is to cluster those cells. So we visualize this here by different colors, and now we have to ask, what do these colors actually mean? And so we basically go through the genes in our panel and check what they mark. Going back to the T cells, we can, for instance, now also on this UMAP overlay this with the expression of CD8A. So the color now denotes for each of the individual cells how much CD8 is expressed, and we can see that there are certain areas that where cells do express CD8, and this corresponds to the orange and brown areas over here. So these clearly are the CD8 T cells. And we still have to find out what's the difference between the orange and the brown clusters here. And the solution comes with looking at Ki67. So we can clearly see that we have a proliferating CD8 T cell population here, whereas these guys are not proliferating. And so this was just exemplifying like how we do that for 2 clusters. And I don't have time to go through all the clusters and how we named them here, and but we end up then being able to actually characterize what these populations are. So we have CD8 T cells here. The green ones are CD4 T cells, violet CD4 proliferating T cells. There's a macrophage population down here. This is the actual skin, the actual skin tumor cells with some of these proliferating. We have some dendritic cells here, some B cells here. There are some fibroblasts, and there are some endothelial cells over here. So this is our first draft of a cell type annotation. We're still working on the details of that, but now, given that we have that, we can actually go back into space, right? Because this is what you would also get out of a standard single-cell sequencing experiment from this tumor, but we went for a spatial, and so what I show you here now is the same cells, actually with their spatial positions. And I color code them by their type. And if we just do it like that, I think one of the first observations that we can make is that there's no, that cell types are quite mixed. And that might not be totally surprising, but still was important for us, whereas there are some areas where maybe here it's more bluish, so there's a certain cell type here, or some fibroblasts seem to be over here. Right, so cells in general are mixed. But we can look at now individual cell types more precisely. And for instance, here, what I plot is the density of the cluster 11, which corresponds to the proliferating tumor cells. So we characterize them based on their transcriptome, right? And now we can look where they are located in space. So the proliferating tumor cells are here now color-coded by their local spatial density. And we can see that there are certain areas of the tumor or of the tissue where tumor cells proliferate more, and other areas there are less. And then we can start to try to correlate how this relates to other cell types. And for instance, we can look at the CD8 T cells, and we can actually find a kind of mirrored image where the density of CD8 T cells seems to be low in the areas where we have lots of proliferation, and vice versa. And so I think now we're ready to go into like characterize better the individual areas of this tumor, and we are only starting to do that. But I think this already demonstrates that this technology allows a quite detailed characterization of what's going on in this patient. For now, that's where I'm going to end, and I just want to summarize again. I wanted Rebekka and me showed you that we established a workflow to characterize the spatial distribution of immune cells and actually all other cell types in serial tissue sections, and we used with transcriptomics, the xenomics approach, and also proteomics with the Maxima. And for now, as Rebekka already mentioned, what we saw is that the brain metastasis was highly infiltrated by immune cells, and especially T cells then contain a fraction of proliferating cells, which also shows some spatial heterogeneity. With that, I'd like to end our talk. I'd like to thank all the collaborators and the facilities involved, and of course also our funding. And yep, I think we're happy to take questions. Thank you very much.
Thank you very much, Fabian and Rebekka. So there's open for questions. Anybody first? Fabian, maybe a very simple question. If you take 377 genes, 1 million cells, this one section, how big is the data?
In terms of size, it's like, I think that it's not that it's like altogether it's 50 gigabytes, and I think it's like could be like go down to 10 or something. So its size is not so much of an issue because it's not so many genes, right? So if you would go for full transcriptome, it would be like nearly factor 100 bigger. So that's still doable. But maybe like regarding to that, what if you work with standard single cell pipelines, definitely things become quite slow. So we like computation time sometimes is an issue. So that's challenging on the data analysis side. Yeah. So we sometimes run. So some of my computations now did run for 12 hours, and it's too long to like interactively work with.
Are there in the internet? Is there somebody checking for questions? No. Okay. That's good. Any questions from the floor? Because I have more, but another question. Yes, please, Stefan.
Yeah, two questions. How were the genes selected? And second, how sparse is the data? So how many, whatever you call that, dropouts you have per cell? Or how many of the genes you see per cell, let's say it this way?
Should I go ahead?
I think so.
Yeah, the genes are now were selected with they were selected by 10x. Basically, they provide some panels, and I think was this a breast cancer panel?
It was a pan cancer commercially available kit. They have a lot of space for customized, but in this case there was no customized gene included, so it was just what they offer, the pan cancer kit. Yeah.
And yeah, and regarding dropouts, I think from the what you could see, so you have about 100 transcripts per cell, and that's the mean number of transcripts per cell, and I think you detect on average you detect something like 25 genes per cell or something. So I don't exactly know the number. Might be like but approximately like that. So 10% of the cells in the panel are actually detected. So it's still sparse, but okay, I'd say.
Along these lines, Fabian, and it's early days, I know that, it's also not so much in other centers, but there could be a disadvantage because of that sparsity that we're detecting only highly expressed genes. Yeah. So have you looked into the genes that you are detecting of the 377 gene panel of those, and you can rank them based on bulk transcriptomics or isolated cell transcriptomics, where you know the expression level ranking per cell type, whether you have actually this bias that you more or less mainly identify those that are really highly expressed, because that would be a limitation still then for spatial.
We didn't do that here, and I'm not sure that we now would have. I guess we would need in this case, right? This is a quite special tissue, so I don't think we have the biotranscriptomics of this particular patient. I mean, what you on the other hand, what you would do ideally if you set up such a spatial transcriptomics experiment, and what we now start to do, and we will get new results on that soon, as if you actually decide you can design your panel in such a way that you make it more sensitive to lowly expressed genes and less sensitive to highly expressed genes to balance this out by adjusting the number of probes. But yeah, so we there will be like during this year we will get samples from other tissues where we then will have single cell transcriptomics and spatial transcriptomics at the same time, and I think then we can really look into that, and that's definitely interesting, I think, to then compare both.
Yeah, thank you very much. There's much to be learned, I guess, and further developed. Are there any final questions from the audience? So last one, Nicolas.
What is really fantastic in what you are presenting is the fact that you would have protein and also RNA. How big is going to be the overlap of protein and RNA? It's going to be 10 protein, 20 protein?
So I think that is what we want to figure out. I think we just have now the issue with the registration because that we don't have everything on one slide, but we have serial sections. But I think we are not trying exactly to put them one of each other to find out how we can somehow get an estimation, what gives us the transcript and then the protein. Yeah.
But maybe the question also was like, how many proteins do we have that kind of the number of molecules on the protein level, right? The number of genes that we have is like.
So how many proteins we can stain?
Yeah.
So we have now 70 proteins in the panel that we can stain, and of course, if the transcriptome data are available, we will do the overlay.
Yeah, and like many of them are in the transcriptome, but not also. But maybe it's like 50 or 40 that we have in both panels.
Yeah.
Thank you very much, both of you, and maybe you can still watch a little bit the next talks. Thanks to Dresden. Bye-bye.
Bye. Thank you.
So we come to the next center, which is Franziska Kimmig from Kiel. So we go north, and Franziska will tell us about population-based analysis of longitudinal variation in peripheral blood transcriptomes.
I will just share my screen. Hi, thank you for the introduction and for the opportunity to present here today. So I'm stepping in for my colleague Neha Mishra, who sadly can't be here today. And the project that I will be presenting is a project that we both are working on, but most of the analysis that I will show today were actually performed by Neha. And I would like to talk about longitudinal variation in peripheral blood transcriptomes. Now, as you all know, peripheral blood transcriptome analysis has been very frequently performed in research due to its high accessibility, minimal invasiveness, and patient's acceptance. But RNA signatures derived from such analysis undergo extensive temporal variation, even in a healthy state. And this is influenced by numerous individual factors, including genetics, changes in cellular composition or cellular life history of environmental exposures. And to distinguish true disease-associated alterations from these natural fluctuations and to robustly identify biomarkers, we need to have knowledge of this transcriptional variation in a normal state. And this has already been investigated by other groups in the past. For example, in the context of differences between populations or different lifestyles, as well as with regard to technical variation introduced by different laboratories. But a comprehensive reference of the temporal variation within individuals is still missing. And with this project, we would like to address this need. So we want to identify dynamic changes in gene expression patterns across time in a normal state in the population, and also assess the magnitude and the different sources of this longitudinal variability. And moreover, we want to identify key genes and pathways with a high inter or intra-individual variation, and then set these into relation to different clinical and demographic variables. So to achieve this, we are using the following cohort. So we have 333 individuals, and we collected blood samples from these individuals at regular intervals over the course of a year. And then we used these samples to perform bulk RNA sequencing. So we have two different sampling seasons, cohort A and cohort B, with 267 and 66 individuals respectively. And in each of those cohorts, the individuals gave blood at 3 different time points, which lay roughly 2 to 3 months apart. So cohort A had its first sampling time point in winter with 1 in spring and 1 in summer, while cohort B started in summer with 1 additional sampling time point in autumn and 1 in winter. And these multiple samples from the same individual allow us to investigate temporal changes within individuals, as well as differences between individuals. So as you can see, cohort A is larger than cohort B, but the ratio of male to female participants is quite similar. And we also don't observe any significant differences in the age or the BMI of the participants between the two cohorts. So first of all, we performed dimensionality reduction in the form of principal component analysis to identify, to assess the global effect of different technical and demographical variables on our data set. And we could observe that the first principal component reflects variation introduced by different cohorts, as well as different sequencing runs in our data. And the effect of age and sex were mainly reflected in the fourth principal component. So we can see here the gradient from young to old participants. And we also see a difference between male and female individuals in our data set. And this correlation plot gives an overview of the association of the first four principal components with some additional variables. And we see strikingly a very large association of individual with all four principal components, and they account for around 48% of the variation in our data set. And we also see an association of annual season with the first four principal components, and also an effect of the time of sampling, so when the sample was taken during the day, and also the BMI. And to assess these different sources of variation in our gene expression data more formally, we next performed variance partition analysis. So for each gene, we computed the proportion of variation that could be explained by different variables listed here on the x-axis. And in this plot, you can now see the variance explained for all the genes for every variable. So each dot here represents a gene. And to the right, the average variance explained by the different variables is depicted. And here we see that individual accounts again for most of the variation compared to the other variables. And this is followed by the variance explained by different measured cell types. So we have here neutrophils and lymphocytes, and then with a little less variance explained monocytes, and also eosinophils with some highly specific genes here. And we can also see the effect of different technical variables on our data set. For example, the sequencing hunt with a high effect, but also cohort and again, time of sampling here. And then as this is a population cohort, so we are interested in the natural variation over the course of the year when people are living their lives. We have also individuals that reported an infection at the time point of sampling or a vaccination shortly prior to sampling, and we see also this in our data set. And then we have the usually demographically assessed variables that also play a role, here age, sex, and BMI. And then lastly, we have different measures taken from our blood samples, and these also affect the transcriptome, including triglycerides, for example, or CRP. So overall, we found that individual and cell type proportion are two major sources of gene expression variation in our data set, and we wanted to investigate both of these further. So we have now looked only at the average expression explained by each of these variables, but we can also focus specifically on genes that are highly affected. So here you can see a few examples of genes for which high variance in their expression is explained by different cell type proportions. So we have on the x-axis for all the genes, the proportion of variance explained by different variables. So variance explained by individual in blue, neutrophils in green, monocytes in yellow, lymphocytes in orange, and eosinophils in purple. And then unexplained variance is depicted in gray here. And here, interestingly, we can observe that for these top lymphocyte genes, more variance is explained by individual compared to the other cell types. And we can also focus on the genes themselves that we see here with high variance explained. And we, of course, find that they are enriched in different cell type markers. We also can detect genes that are involved in the regulation of cell activation and differentiation. And we also can see genes involved in cytokine production. And then we also wanted to investigate genes for which a high variance could be explained by the individuals. And these included, among other things, genes that are involved in T-cell receptor signaling. And I have selected 2 examples to show you the expression in our data set. So we have on the x-axis the different individuals from both of the cohorts, and then on the y-axis you can see the expression of these 2 HLA genes in our data set. So for each individual at each of the 3 sampling time points. And here, for most of the individuals, the expression of these genes is quite similar between different sampling time points of the same individual, and differs noticeably between individuals, but there are also a few outliers here. And when we now color the samples by reported infections or vaccinations, we see that these outliers can actually be explained through these events. And then we also wanted to investigate inter and intra-individual variation. So for each gene, we computed the proportion of variants that could be explained by longitudinal changes within individuals, so the intra-individual variation, as well as differences between individuals, so inter-individual variation. And here we can see that intra-individual variation is actually much higher compared to inter-individual variation. And we can plot these for every gene. So we have here on the x-axis variability within individuals, and on the y-axis differences between individuals. And we can see here that there is a set of genes that is highly conserved in their expression between different individuals. And these include also the T-cell receptor signaling genes that we found before with high variance explained by individual. And then on the other hand, there's a set of genes with high temporal fluctuation within individuals and low difference between individuals, and these include several ribosomal genes. And these 2 measures of inter and intra-individual variation are combined in the intra-class correlation coefficient. So this is a statistic that is usually used to describe how strongly units of the same group resemble each other by comparing the variance within and between groups to each other. And in our case, we can use it to measure how consistent the expression of a gene is. So is the gene expression more similar between different sample time points or from the same individual or between different sampling time points? So a value close to 1 would indicate a very conserved gene expression, whereas a value lower than 0.5 of the ICC would mean that the gene expression varies more within individuals than between. And this is the distribution of the ICC for the expression of all protein coding genes in our data set. And we see here that around 85% of the protein coding genes actually vary more within individuals than between. But we can also see a set of genes here that is highly conserved in their expression between individuals. And then to show you how ICC actually reflects inter and intra-individual variation in our data set, I have again selected a few examples. So these are 2 examples for genes with a very high ICC of 0.94 and 0.83. And we have again the individuals of both cohorts on the x-axis and then the expression of the gene on the y-axis. And these gene expression patterns are very similar to the genes with high variance explained by individual. So we see here a very similar expression between the different sampling time points of the same individual, and a significant differences between individuals. So we can almost distinguish different individuals based on the expression of these genes. And this is not the case when we look at genes with a low ICC. So here we have a ribosomal gene with an ICC of 0.21, and we immediately can observe a much higher temporal fluctuation of the expression of this gene in individuals. And next, we performed an overrepresentation analysis to identify biological processes that are enriched in these genes with high or low ICCs, so high inter or intra-individual variation. And this ridge plot depicts the distribution of the ICC of the genes that these terms are enriched in. And when we now focus on genes with high inter-individual variation, we see that they are mainly related to biological processes that are in connection with immune response. So we have here, for example, eosinophil chemotaxis or peptide antigen assembly. And we see that there are some genes that are very conserved within individuals that contribute to these terms. And on the other hand, when we look at gene sets with high intra-individual variation, we see that they are mainly associated with housekeeping processes, such as mitochondrial translation. And until now, we have mainly focused on the variability of gene expression, but transcriptional variation has also a different source of variation. It can manifest in differences of the splicing patterns of a gene. And we now wanted to assess the contribution of these two sources of overall transcriptional variability. So on the one hand, changes in expression, and on the other hand, changes in the transcript usage of a gene. And to achieve this, we've used a model developed by Gonzalez-Porter et al. So this method computes for each gene a value between 0 and 1, where 1 indicates that the gene varies mostly in its expression, and a value close to 0 means that the gene varies more in its splicing patterns compared to expression. And this is the distribution, again, for all protein coding genes in our data set. And we see that around 60% of these genes vary more in their expression as opposed to their splicing, with one set of genes here, close to 1, that is almost exclusively varies in their expression. And but there's also a significant amount of genes, around 40%, that vary more in their transcript usage than in their expression. And also in this case, we performed an enrichment analysis. So to investigate the biological processes that are enriched in the genes at both ends of the scale. And here we found that genes that vary mostly in their splicing are associated to different inflammatory processes, such as neutrophil chemotaxis and interferon beta production. And on the other hand, genes that vary more in their splicing are associated again to different housekeeping processes, such as DNA repair or transcription. And next, we wanted to investigate the relationship between inter-individual variation, and the proportion of variation that could be explained by expression as opposed to splicing. So we separated all our genes into 3 groups: the genes with high inter-individual variation, mid and low inter-individual variation, and then we visualized the proportion of variation introduced by differences in expression levels. And here we found that genes with high inter-individual variation actually vary in most cases more in their expression than in their splicing, whereas genes with low inter-individual variation vary mostly in their splicing patterns. So, and then lastly, as our samples were collected over the course of a year, we wanted to see if we could detect the effect of season on our transcriptome data. So the samples in cohort A were collected between February to August, so in Belgium, so they would reflect the winter to summer transitions in Northern Europe. And on the other hand, samples in cohort B were collected from August to February. So these capture mainly summer to winter transitions. And we now want to analyze both cohorts together. So we tried to align them somehow. And we did this by matching the samples collected in winter from both cohorts and also the samples collected in summer. And then we interpreted spring and autumn basically as intermediate season samples. So this is how the alignment worked. We have cohort A and then the reversed cohort B. And this enables us with a greater power to investigate and detect genes that change in their expression and in their transcript usage from summer to winter or vice versa. And using this method, we detected a total of 4,269 genes with changing expression between summer and winter. So out of these, 2,057 genes were upregulated in winter compared to summer, and 2,212 genes were higher expressed in summer than in winter. So also in this case, we performed an enrichment analysis. And here you can see the heat map of the expression. So we have averaged over the individuals to get a clearer view. And we found that genes that are involved in the response to interleukin-7 production, to the cellular response to interleukin-7, as well as that are involved in interleukin-12 production, they are in general upregulated in winter compared to summer. And on the other hand, genes associated with hydrogen peroxide catabolic process are upregulated in winter compared to summer. And we were also very happy to see that some genes that are involved in the entrainment of the circadian clock are also differentially expressed, and that this is detected in our data set. And then next, we also try to detect genes with different transcript usage between the two seasons. And we found 1,193 genes that exhibited such season-dependent splicing patterns. So out of these, 741 exhibited differential transcript usage as well as differential expression. And on the other hand, 452 of these genes only show changes in their splicing patterns without presenting significant differences in their expression. And here you can see a few of the most significant examples. So to summarize, we observed that individual and cell type proportion are two major sources of variability in gene expression. And we also found that most of the genes varied more within individuals over time than between different individuals. And here, inter-individual variation was mostly associated with different immune responses, whereas intra-individual variation was mainly related to different housekeeping processes. And then we also found that transcript usage actually contributes significantly to transcriptional variation. And this variation is mostly within individuals and not between individuals. And also we could detect a seasonal effect of the seasons on gene expression as well as transcript usage. And of course, there are still a few things left we would like to investigate. So in the future, we would like to analyze if we can detect the gene signature of different food metabolites or cytokines in our data set. So this would be, for example, to see if we can detect a higher signature of the presence of vitamin D in summer samples compared to winter samples. And then secondly, it is well known that the cellular composition changes between seasons or over the course of the year. So we are wondering if the genes that we detected with this method were only detected because of these compositional changes. So we would like to perform cell type deconvolution to be able to include these imputed fractions in our linear mixed models and see if we still detect the same seasonal genes. And then we would also like to perform gene co-expression analysis. So to detect longitudinally co-regulated gene modules and also set these into relation to different clinical and demographic variables. And then lastly, I showed that we also have some samples from individuals who reported infections. And in most of these cases, only one of the 3 sampling time points were actually had an infection. So we have here the possibility to investigate the longitudinal signatures of bacterial and viral infections in our data set. So with this, I would like to thank everybody who is involved in this project. So a special thank you goes to my supervisor, Philipp Rosenstiel, and Neha Mishra and the whole systems immunology group. And then also I would like to thank Jeroen Reyes and his group from the Center of Microbiology in Leuven, who provided these samples for our analysis.
No, thank you very much, Franziska. I think very important to those that want to use transcriptomics for clinical biomarker development. Are there questions from the floor? Maybe I start with one is concerning the housekeeping genes. You showed one example, and you also the range of the variation. In immune genes, we know that, you know, by infection, but also by chronic diseases, you will have high effect sizes in some of these genes. But when you take now, for example, an RPL9, which fluctuates between 8 and 14 in the population, and you have still like these are log scale, I guess, right? In the expression level with one individual quite highly. Do you think there is diseases that this fluctuation could be even higher, or is this then just something we have to dismiss when it comes to biomarker development for diseases?
That is a good question. So we actually have a few patients who reported diseases in our data set, and this plot is including these patients. So we have some with chronic inflammatory diseases and others. So, but we are analyzing at the moment also our data set subsetted to all the healthy individuals, but we didn't specifically look at ribosomal genes, for example. So this would be a good suggestion to do in the future.
Yes, please.
I had a question because I was also interested in these graphs that you showed as individuals. One other graph that you showed, there were actually multiple steps in this curve. So you could, this one, right? So instead of having a continuum over the individuals, you kind of have two states, right? Low and high. And in the upper of the graphs, you actually have multimodals, different states. Is that something that you could use to classify genes in terms of whether it's a continuum, whether it has separate states, separate plateaus?
That is a good question. So we haven't thought about it yet, but I think it's worth looking into. So I don't know why that could be the case.
Fabian, can you hear us?
Yes.
Please, go ahead.
Well, I've been running. So, if I understood correctly, that like in your trial, was like between individual measurements, there were like a couple of months time, right? I was wondering what would be like the persistence time of your of the transcriptome. Like, if I would get the transcriptome the next day, would I expect similar kind of variation, or how fast do you think this would change? Is there anything known?
That is, I don't know actually. So we had to make this decision when we planned the data set, if we want to have more frequent samples from the same individuals or different individuals, and then less frequent sampling. And I think it's both worth looking into, but this helps us to give an overview at the beginning. And then, of course, more frequent sampling is also needed to assess the extent of intra-individual variation that we could observe here.
I might add, Fabian, because we have some data where you have closer sampling, you know, where you cannot see any seasonal aspects, but there, if you're not developing an infection or anything like that, and if you're not changing your lifestyle very acutely, for example, you're not getting drunk the night before, then the transcriptome stays rather strictly similar in one individual over a couple of days. But if you are going outside of your normal life, this is seen.
Mm-hmm.
Yeah. And infection is, for example, we see many infections already 2 days before they really start clinically. Yeah. So that is, on the one hand, for biomarker development is an issue, because we even have to ask people later on back, did you develop shortly after the blood take something, which complicates things? And we have to ask people to say what else has happened. And last question for Franziska, the design to see seasonality, I understand, but there's still a difference between springtime and fall. So you could also keep it like longitudinal, just take the summer as the beacon where you combine them and see what is the difference between autumn and fall, no?
Mm-hmm.
I think right now you're not just or I missed it, maybe you distinguish.
Yeah, no, we have basically align spring and autumn.
But it would be nice to see, because you know, there's a difference between getting more light and different food than reducing light and getting different food again.
Yeah, that's true. Yeah.
And what I would be interesting also, last thing is, I think your outlook food intake, you might know we did a study in Tanzania where we looked at that, and also looked in rural areas versus urban areas, which of course is in Africa much more different than it is in our area, but there are differences where people live in their habits. And it would be nice to see whether you could do something like that in your study. I could imagine, my hypothesis would be, if they're coming from Belgium, it doesn't matter whether you have a city or a rural area, but in Africa, it's a huge difference, because the intake of food and the habits between somebody living in a city in Africa and a rural area is enormous still.
Yeah, thank you.
Last question, or two more? Yeah, I think we have. Thanks.
Thank you. There are two questions from online audience. The first question is from Suzanne Montamini. You mentioned that you were almost able to identify individuals based on the expression of certain genes. Do you think that this has implications for processing of gene expression data with regards to data protection regulations?
Oh, I don't know. Sorry.
No problem. The second question is Konstantinos Vargagiannis. I hope I don't ruin that. Considering the time of sampling within the day, what is the same for all was it the same for all participants? Is it possible to have this type of intra-individual variation within your data too?
Yeah, so we have actually a wide range of time of sampling during the day. So I think the earliest samples were taken around 6 a.m. in the morning and the latest at 8 p.m. in the evening. And but most of these samples fall in the range between 6 a.m. and 10 a.m. And we were also thinking if this was responsible for some of the intra-individual variation in our data set. So what we are doing at the moment is, like with the healthy individuals, we are subsetting to the samples taken in the morning, and then we repeat all our analysis. And until now, for most of these things that we have analyzed, we don't see a real significant difference between focusing only on the morning samples or the whole data set.
Last question from here.
Yeah, thanks. Really nice work. I was wondering, did you check genotype differences, especially inter-individual differences? Especially EQTLs, for example, could that explain many of the inter-individual differences?
No, we haven't done that yet.
Okay, thanks again for this very interesting talk, Franziska. We now come to the third talk from Kerstin Ludwig from Bonn here, and she will give us the update on a NGS-wide network study, still from Decoy, where I think we generated one of the largest WGS data sets for COVID. Not knowing that this happened, when we started the project, we thought we're giving the network a certain number of genes to be sequenced, and we're not sure whether this is going to be in the high range of competitors or low range. And Kerstin will tell us where we landed up and what the results are, right?
So thank you for the invitation to speak here to you today. So this will be, I think, the only talk about genetics in that session. I would like to invite you to join with me on join me on the journey that we did in COVID-19. And as Joachim mentioned, I will also present some genome sequencing data, but that's to become at the latest or at the end of my talk. So I keep you suspense until then. I would like to just remind you about something that you all know of, just to keep you back or get you back on the numbers a bit. So the SARS-CoV-2 pandemic has hit us hard globally. And even though we are not testing anymore and do not follow up on that in the acute infection at least. So I'd like to remind you about the numbers, right? So we had approximately, or at least about 770 million infected people in the world. And this included also 7 million people that died from COVID-19. And of course, the situation got a bit better in terms at least of the fatalities when the vaccinations entered the world. And you see in the numbers of the vaccination that has actually contributed a lot to the fact that we are not talking about these huge death numbers anymore. But of course, given that long COVID came around, the acute infection of COVID will still be keeping us busy also from the genetic side in the next years. So just to remind you about when we talk about infectious disease and in particular COVID-19, I'd like to remind you that we deal with different phenotypes. So at first we do have differences in your susceptibility towards the infection. So I don't know whether some of you still know people that have not been infected at all, although probably everyone has now gotten exposed to it. So there is a couple of people that are resilient or resistant to infection, which is nice to study. But of course, the majority of us got infected at some point during the pandemics. And then there was the question of, okay, how do your body behave? Do you have an asymptomatic infection? Do you have a mild infection? Or there were also a lot of people that had, of course, a severe infection. And that was basically the acute infection period that we worked on during the first years. And now we see the picture that some of the individuals, or most of the individuals, recover pretty mildly. They don't have symptoms that are long lasting, but others actually do. And at the moment, we still don't know how large this number in the end will be. And for these called post-acute sequelae of COVID or of infections have been known for a number of infectious diseases, but they have not gained so much attention because of the fact that the number of individuals that were infected was rather low. But with COVID-19, that has now changed because of the pure and sheer number of infected people, we will also see a lot of people that have to deal with long COVID on the longer term. So, and if we want to study the reasons for that, we of course need to look at different multifactorial aspects. There's of course a lot of things that we can attribute to the virus itself, your viral load, your strain that you got infected with. Then we have the normal suspects like your age, your lifestyle, and also your gender or sex, of course. And then we have a lot of things on the host side. So the variability on clinical measures that you all know of. You can, of course, know the body mass index or obesity is one of the strongest predictors of severe acute COVID. And then many of them are, of course, mediated by genes. And then we have a set of traits that we don't know yet that confer risk for severe COVID, which are also genetically mediated. So if we understand the genes that contribute to our susceptibility and our acute infection, it will probably also help us to understand the post-acute infection etiology. And this is actually where the journey of the COVID initiative in Germany started. And it became clear pretty early in the pandemic that we need huge cohorts to get some ideas about the genetics. So there has been, and I'll show you a couple of those studies that have been established very early in the pandemic with the idea to study genetics. They differed a bit in their informed consents and the way they were structured. So not all of them had, for example, consents for exome or genome sequencing. Some did, of course. And in Bonn, just as an example, we here also founded one of these studies, which is the Bonn study of COVID genetics, where we started to look into susceptibility in acute infection. We did that on a population-based scale. So we asked just people from the population to provide us with questionnaire information, but also with a saliva sample. So that was actually pretty practical when the people were all in isolation or in quarantine. Then they could just send us saliva samples and do things from home. And we also included recontacting options at that time. So which means we could actually go back to the patients or to the probants and ask them for more information when it is needed. And of course, that's now pretty handy when we talk about long COVID, because we can now recontact the people and ask them for longitudinal information on their disease. So with many of these studies, and actually many of the colleagues that contributed to these studies are here, we then run different analysis to study the genetics. And for those that are not so much familiar with the genetic architecture of multifactorial traits, this is a picture that is often shown to show you actually that we have to look at different angles. We have to look at rare variants, we have to look at common variants, and we have to do so using different technologies Of course, eventually genome sequencing would be the one that gives us all, in particular, if we then include long read aspects of it. But financially at the moment, it's not really possible. So you often do like a joint approach. You do error-based genotyping to study the common variants. You take sequencing approaches to study the rare variants. And ideally, you take exome sequencing to get all the protein coding variants. But then you still miss the 98% information from the non-coding regions. And that's where genome sequencing comes in. I know that the topic of today is not about common variants. So I would just like to highlight that the efforts we did in Germany were also, of course, accompanied by many efforts worldwide. There was a huge initiative in the beginning, putting all genetically interested people together, which is the COVID-19 Host Genetics Initiative, with the idea to study the common variant aspect of COVID-19. And this was an increasing cohort with with regular data freezes. And the latest data freeze was actually published in September 2023. And that study included 219,000 cases, 3 million controls, 82 studies, and 34 countries. So it was a huge global effort. And just to illustrate it to you, the number of authors on that paper was about 4,000. And that's the picture. So these are the loci that we currently know of in terms of common risk variants. And I won't go into details of that, but I always find that nice to actually put it to show you that we actually have this common risk background that also contributes and shapes each of us personal profile for COVID-19. And there's actually one locus, which is on chromosome 6. It's called FOXP4 locus. And this locus is now also the first one that has been recently identified as a susceptibility locus for long COVID. Okay, but as I mentioned, we are talking about sequencing today. And so I will present you two studies that we did to study rare variants or low frequency variants in severe COVID. Actually, 3 studies. The first one is not such a large one, but of course, the best, or let's say the most informative way right away is actually when you study extreme cases. And this can be done in, for example, a clinical diagnostic setting. And this is work that was led by Axel Schmidt, who studied together with many people also here in the room, a 3.5-year-old girl that was a member of a consanguineous family. And this girl, she had a very intense and very, in the end, also fatal disease outcome after COVID-19 or after SARS-CoV-2 infection. She actually died of multi-organ insufficiency 62 hours after she came to the hospital. It's one of the very few fatal children with fatal outcome in Germany. So when there was an exome done, what we found, or what Axel in particular found, was a TBK1 splice site variant located four base pairs within exon or following exon 16, within intron 16. And that girl was homozygous for that variant, and her parents carried that heterozygous. And I don't go into details of that particular finding, but just like to highlight that this TBK1 deficiency was then, through other studies worldwide, actually implemented as a new autoinflammatory disorder called TBK1 deficiency, and it's now also listed in the official inborn errors of immunity list. When you want to study, when you want to look at what contributes to severe COVID on a more like population scale and not in individual cases, you would ideally like to have large cohorts of very severely affected individuals. And one can say, luckily in Germany, we did not have these super heavy waves that were seen in other parts of the world, for example, in Italy. And I think many of you remember these pictures that were transmitted to the world from Italy in particular, but also from Spain, where they had a huge and very early and very heavy outbreak of COVID-19. You see in the picture below, it's actually 30 days after the first report of a case in Italy, when the entire country was really heavily affected. And Andre Frankel's group from Kiel, they had, through collaborations, they had access to a large cohort from that region, and they had a lot of individuals that were very severely affected. And this was actually the first, when they analyzed that with error-based genotypes, they were also able to identify the first risk locus from the common variant side for COVID-19 just 3 months after the COVID-19 outbreak, showing you that when you have established collaborations, you can actually be pretty fast with the genomic technologies we have. But that cohort was also really interesting for rare variant analysis. And this is work that was done by Yannick Bos from our team. And we used actually this cohort for studying rare variants, but we could just only do targeted sequencing because we did not have informed consent for exome-wide analysis, unfortunately. So we had access to 1,770 cases and about 5,300 controls from Italy. And we sequenced it in the West German Genome Center in Bonn using molecular inversion probes technology. And we had to select candidate genes for that study. And these are the candidate genes that we selected at that time. If I look back, I would probably now change the list a bit. But we included the early GWAS loci, so genes at the common variant site. We also included genes from the diagnostics. You can see TBK1 right on the right-hand side. And we also included a lot of genes that we had evidence for from the literature. One striking gene is, and it's on the diagnostic and literature side, is TLR7, which was one of the first monogenic forms identified from a group in Nijmegen. So what did we find? So first we looked at single variants, right? This is of course challenging because single variants, just in terms of power, are very likely not to come up at frequencies to get them at a certain frequency that will allow you to put them statistically significant. But we had two actually very interesting findings on the single variant side in low frequency variants. All of them had a minor allele frequency below 1%, but they were more often reported than just singletons. So the first one is a variant in the TBK1 gene again, which it has this variant is a missense variant. It has a pretty high cut score. The Revel score is in the middle. And in our study, we observed that with an odds ratio of 30. But just because of the power of our study, it did not reach, it was only nominally significant. We did not get it significant when we correct for multiple testing. But there's another study out there that has exome data available for COVID-19, where you can just look up individual variants and then you get odds ratios and p-values as well. And this is the Regeneron cohort in the US. And when we looked up that variant there, we actually found an odds ratio of 24.2 for that variant, and combining both analysis yields a pretty significant p-value, but also a pretty significant odds ratio for a single variant within the TBK1 gene. And the second finding is a population-specific variant that is specific to Spain. It is also a missense variant, this time in TLR7. And again, the cut score is pretty high, and we have an odds ratio of about 5. As this variant is a population-specific variant in Spain, it was not reported in the Regeneron browser just because of it's not available there. So we still lack independent support for that variant. So what can you do if you have large cohorts and you look for rare variants, but you don't really have the power to find individual variants? You can, of course, collapse your variants into across genes or across gene sets, which is generally referred to as burden analysis or combined analysis. And for those that are not familiar with the concept, what do? You basically take all individual variants that you find in individual genomes per gene or per gene set, and then you filter for frequency, but you also filter for potential functional effects. There's a lot of limitations, of course, associated with that, such as the lack of annotation. You often do not know whether a missense variant is actually benign or pathogenic. And then you have population-specific variants. But with all these, keeping those in mind, you still can do filtering, and then you basically sum up the variants across a gene and say, okay, if there's at least one variant in the gene, no matter what position it is, but it meets my certain criteria, then I say, okay, it's a carrier, and the others are not carriers, and then I can compare the frequencies of those. And with this concept, you can increase your power because you can aggregate the rare variants into individual observations. And at the gene burden level in that study, we actually found one gene that's was really striking, which is again TLR7. So deleterious variants in TLR7 were super highly enriched. And you see several measures in statistical significance here, which means that we perform different analysis in terms of the severity, in terms of subgroups, like clinical subgroups of the patients, but also in terms of the variant categories we included. So again, TLR7 is the only one that was really striking. And when we used the clinical information that we had for the patients, and that was also very good because we had this clinical information, we also could actually see that the fraction of carriers with a deleterious TLR7 variant was the highest in those individuals that did not have other clinical risk factors. So it seems to be completely, not completely, but it seems to be an independent risk factor that is not captured by, for example, hypertension or body mass index. That's the numbers on the right side. At the gene set burden, we took all our genes that we had. It was only 55, but we could classify them into certain groups. For example, those involved in interferon signaling, those involved in chemokine receptor signaling. And all those results were only nominally significant, but the most significant finding was, again, for deleterious variants in the gene set of immune deficient genes or inborn errors of immunity, which is not surprising because TLR7 is in that list. So that, of course, drives the signal. But then we excluded TLR7 just to account for that fact, and we still see a significant finding. So apart from TLR7 and probably also TPK1, there's some other genes of the inborn errors of immunity where individual deleterious mutations are likely to increase your risk for severe COVID. But then we also did some subgroup analysis, and that included, for example, the analysis of males and females, as we all know that there are differences in their severity. And what we found very striking, I did not expect, was that we found an enrichment of deleterious variants in the gene TLR7 in females. Why was that surprising? Because TLR7 is an X chromosome gene, and if a woman is heterozygous, and it escapes X inactivation in immune cells. So if a woman is heterozygous for mutation, it would basically still have one functional copy. So you would basically have the same risk as a male person. So it was pretty surprising that we see an enrichment of female heterozygous carriers in severe COVID. And we saw that actually across diverse measurements of variant effect. And when we looked at it in a 3D space of TLR7, we actually saw that they pretty much clustered in the dimerization interface. Why is that important? Because TLR7 works as a dimer. So if you have one not functioning copy and one functioning copy, it might actually be that they interfere with each other. So you while you have 50% gene dosage, you might have 75% of wrong dimers of TLR7, and that is very likely that is a suggestion. We have not followed it up in the lab yet. Is that there's probably in the women a dominant negative effect rather than a loss of function, as we observed it in the males. What I would like to point out that here now is that the fact that we need to incorporate to improve our variant interpretation in the future, we might definitely look for more information from the protein side. And I think Axel did some work on that, but there's also recent new tools like Primate 3D, which can be probably very helpful in improving our variant interpretation. Okay, now I'm suspense. Now we're talking about the study that Joachim mentioned, and that was again led by Axel. And I think it's a super nice illustration on how a network of people that work together can actually achieve very nice things. So these are it's a genome sequencing study, which was mainly run at 3 genome sequencing centers, which was the West German Center Bonn, Cologne, and the NCC in Tübingen. And we had gathered a cohort of 1,220 samples. Most of them were hospitalized, some of them were mild. And this is, as Joachim mentioned, one of the very few genome sequencing data sets on COVID-19 worldwide. It is still rather small when we talk about a multifactorial trait, but I think we can still learn some interesting things from the data. So this is the structure of the cohort. So we started off with 1,275 individuals. And after some quality criteria, we ended up with 1,220 individuals where we had really good sequencing data. And then we did some analysis on the population level, because if you want to do more statistical approaches and also in terms of common variants, you need to have a population homogeneous data set. And not surprisingly, that in our case was a European cohort of 1,017 unrelated European individuals. So we have a decoy cohort and we have a decoy Euro cohort. On the right-hand side, you see the age distribution of the different groups. And again, not unsurprisingly, there is a larger the people that have severe COVID tend to be older in average than the people with a mild disease, but we have a pretty good group size in each of those groups so that we can run statistics. Now, the good thing about genome sequencing is that you can not only look at the entire genome, but you can of course also look at the protein coding genes as you would do for an exome. And so what we did is we wanted to see whether we have monogenic conditions among our cases or among the probants in general, and whether we actually can identify monogenic conditions that might increase the risk of severe COVID. So Axel went into ClinVar and took out all the pathogenic variants at certain criteria that are pathogenic or that are causative for a monogenic disease, either in autosomal recessive or in autosomal dominant manner. And while we found a number of diseases or variants associated with autosomal dominant diseases, 77 in total, we did not find a significant overrepresentation or significant difference in those carriers in terms of their clinical characteristics. This was a bit different for the autosomal recessive ones. Here we found only 4 individuals that carried a recessive form. And when we then looked on the right side on their clinical characteristics, we actually saw that all of them were male, all of them were younger than the average cohort, and all of them were at least hospitalized. None of them was a mild one. So the genes are listed there. So these 4 genes are now prime candidates for being involved in severe COVID from a monogenic side, based on, of course, the fact that the carriers had this underlying monogenic condition. And then you can do the same thing also, or you can actually look at the common variants also. And you can do that with the polygenic risk scores. And what we did is wanted to actually see whether the common polygenic risk score is different across different age groups in our cohort. And we generated a polygenic risk score based on the genomic cohort. The reason for that is that the genomic code is completely independent from ours, of course. It's from the UK, and it's whole genome sequence data, so we don't have a technical issue here. We only performed that analysis on the European subcohort, obviously, because that's where you have to be restricted to your population. And what we saw is that we did 2 different groups. The first definition was cases were severe hospitalized individuals compared to all others. And the B analysis is you are hospitalized as when you're a case, and when you're not hospitalized, you are control. So in both cases, you see that the cases have an increased polygenic risk score, which is nice to see, but also kind of expected. But this difference is actually different and significantly different for the different age groups. So when you are the polygenic risk score component in disease etiology is stronger when you are younger than 60 years than when you are older. And that is actually true for both phenotypes. So what we can show here is that the genetics play a role much stronger in the individuals that are severely affected but young, as opposed to those that are severely affected and old. And when you have that data, you can then, of course, also start to look at candidate gene sets again, or at gene burden of rare variants again. And while these results are only nominally significant, there's no genome or no test-wide significant finding due to power. It's worth mentioning that we found a couple of enrichments of, for example, putative loss of function variants in genes of the innate immune system. And although we did all comparisons that we could do, we did only found anything significant in the overall groups or in the males. So again, there seems to be a stronger effect on the genetics in the male groups than in the females, at least in our data. So now we have done a lot of puzzle piece identification and the genetics and statistical side. Now the question is, does it tell us something about biology? Yes, it does. And the really nice thing is that we are now starting to have a large enough sample size or gene size that we can actually see that the common and rare variants we identify, they converge in major biological pathways. The ones that are just highlighted or that are listed normally are those that are from the common variant studies. And then there are a couple of ones that have a green circle around. These are from the rare variants, and they cluster together in gene sets or in the pathways of viral entry and in particular into type 1 AFN response or interferon response, which are this, at the moment the major prime candidates for a genetically mediated biological effect in severe COVID. Okay, are we done yet with COVID? Not yet fully. We are now with that, based on that work, we have now extended our cohort search on the NAPCON cohort, and we have recently gained funding to sequence exome-wide 4,000 individuals of that cohort. For those of you who don't know the NAPCON cohort, that's the National Pandemic Cohort. It's part of the network of university hospitals and has been set up to actually follow up on individuals either on the population base or really in general practitioner clinics or in the clinics. And there are actually 3 different platforms, which means that you have some individuals that have been in the hospital, but they have very, very detailed clinical phenotype data. And others are more on the broader population scale, but then with follow-up time points over a couple of years. So you get a picture of the future involvement also of long COVID. These are the numbers that the NAPCON cohort has gathered genome Germany-wide. We have about 7,100 individuals that have been included in NAPCON through any of the 3 platforms. And not only do they have participated and gave clinical information, there's actually also a lot of omics data that is about to be generated or is currently generated for many of those participants, which now gives us a chance actually to correlate also genetics with the multi-omics data sets. So that's what we are doing at the moment. And that's my final slide. What have we learned from the pandemic and what might be helpful in the future is that if you have an established collaborative network, that can be very helpful. And that is also something that the network university hospitals will now start to look into. So there's plans that this NAPCON cohort and also this structure will actually be evolved into a national study network in which basically university hospitals will be brought together as an infrastructure to then engage into much collaborative work in the case of a next pandemic that might hit us. With this, I'm at the end of my talk. I would like to thank, of course, the many, many people involved in that study. I could not list them all here. As I mentioned on the COVID HDI slide author list, there were like 4,000 people. But I would like to particularly thank Axel and Yannick for their really hard work in their fight with these huge data sets. Thank you.
Kerstin, thank you very much for this very broad journey through genetics and all the international collaborations. I mean, this is the big advantage your field has. I can just say like the immunologists that should have a better saying in this whole thing don't have such networks, and that's a big disaster for pandemic preparedness. We have to work on that and learn from genetics. Any questions for Kerstin? Yes, please.
Yeah, really fascinating work. With the TLR7, I was just wondering, what's the thinking? You know, we have X chromosome inactivation, which will create mosaic cells, right? So how would it be dominant negative?
So you mean how the mechanism?
Yeah, so you're proposing a dominant negative function of the mutation, right? But you will have random inactivation of X.
Yeah, it seems that there's a number of studies that have actually shown that in immune cells it's not so random. So that that there's some. You mean, I know. Sorry, I'm on the right track. You mean that within an individual we have
exactly.
Yeah. Okay. Yeah. So in the end, I can't tell, to be honest. I don't know. So at the moment we only have that concept, but we have a replication cohort which actually showed the same. So at the basic mechanistic function, we don't know. It's just about the 75% and 50% active dimers that we have. As a concept, as we say, it's a potential new mechanism, and definitely have the immunologists look into that from the genetics.
Okay, thanks.
Is anybody from so first from the web?
We have a question from the online audience. Kirsten Becker asks, have you or will you look into structural variants in genome data?
I would love to. That's the plan. The problem here is definitely data transfer problem. It was pretty easy to transfer the VCF data. For joint analysis, it's a bit more complicated and more data, data government problem for the raw files we would need for the joint structure variant calling. So it's on our agenda, but we are not there yet.
Thank you.
Yeah. Have there been similar studies for other respiratory viruses, like host genetic studies, and how do they compare, if there are?
Yeah, so there have been a couple of smaller studies, in particular in the 23andMe data bank, the biobank. The problem is that sample numbers are pretty low because many other respiratory, like the more common respiratory viruses, they are not reported with their that they get. People just say I've been infected, but they don't report any symptoms also about that. So 23andMe did a small study, but they did not find much. And now Regeneron has recently published a study on influenza with more than 1 million participants. Can't say how many of them were cases and controls, but that's what they did. And I think they found 8 common risk variants at the 1 million sample size.
So common means also found in COVID? Or...
no, common means at a frequency level. So, they did a GWAS, they did not do yet a rare variant study. So common, the overlap was actually minimal, if that was the second question.
All right, Kerstin, thank you very much. It was great news. And we come to the last talk of the centers before we have one talk from industry, as it is the tradition. And this is Nicolas Casadei from the center in Tübingen, and he will tell us a little bit more about, I think, technical developments, to say advancements, but also solutions and challenges in sequencing centers. Nicolas.
Hey, thank you so much. Really my pleasure to come in the group and discuss a bit what are we doing in Tübingen, what are we developing at the moment, what we were able to implement. This one, thanks a lot. So the presentation is a bit different from the 3 that we have. We go a bit back from basic research and we go a bit more technically on what was implemented in the last year, what are we currently implementing, and how do we see the future of this method. The very first point I would like to bring is since the beginning of the center in 2019, we really saw that the initiative of the DFG to bring research project in sequencing was very successful. And obviously our workflow needed to scale up due to the amount of projects that came. And we are currently in Tübingen in the core facility, roughly at 50% of the sample are coming from DFG project, 50% of the sample are coming from our usual core facility activities. What is interesting, and I like to highlight it, is genomic field is volatile. And we always need to see a bit what is going to come to us in order to be ready in the next months and in the next year. I think the idea of the GFG, where scientists could come to us, discuss about what do they want to do, how could they implement it into a scientific project, was a really good opportunity to try to see in 1 year, what do we need to scale up, because these kind of projects are coming. When we look, for example, in 2022, we had, at that time point, a certain amount of sample going into long-read sequencing and single-cell RNA sequencing. But based on the consulting that you see on the very right, we knew that we had to scale up these systems because the number of sample coming were at least the double of what we were used to process. And that gives us, when we do research together, a bit the opportunity to see the future, try to scale up, and be ready when the samples are coming. One also point I would like to keep short is I don't want to make showcase of instrument or methods, but these fundings really enable in a center to get multiple technology running in parallel, where we had always the opportunity to test different workflow in the same times, in the same center with the same scientists on the same sample. And this was an opportunity which was really great. And I also think that the scaling up of sample using liquid handler was for Tübingen quite important for reproducibility of data, but also for enabling other colleagues on how to think in larger scale, and also how to bring really this protocol in production scale and not only in a research project. The team typically is composed of 3 different workflows, where on top of a normal administration, we have scientists working in short read, long read, single cell sequencing. And one thing that one can notice in this kind of workflow is our core facility, like other core facility, are really tight with personnel, meaning having one person doing development, one person doing workflow. When someone is in vacation or sick, there is just no production happening. And I guess it's a bit the challenge, and it's a bit the opportunity to work in network in order to feed this workflow and have the opportunity together to make overhead of sample towards centers which has expertise, but also who has capacity to perform specific protocols in order to have research projects being performed on time with good quality. In the talk I'm going to present, most of the data I'm going to show are based on our research focus, which reflect also in the protocol we are providing. And as you can see, most of the protocol which are run into the Institute of Human Genetics are mainly targeted toward applications that could be useful in diagnostics and that are definitely related to human health. Here, most importantly, is as many centers are running protocols in routine, automatized on robot with high throughput, get protocol which we run in research with collaboration with scientists, where we warn them of the pitfall and we work together on solving this pitfall into the data processing, but also into the data analysis. And finally, we have protocols that we call in development. These are protocols we already tried. We are sure that they are working fine, but it's either not really our focus, or we don't see scientists in our surroundings scaling them up, so they are staying in protocol being tested. So when the sequencing center started to scale up, there were a massive request for blood RNA sequencing. We saw from the colleagues from Kiel that data which are generated since a couple of years are being aggregated, and colleagues are trying to interpret them in large scale. And for us, one of the first things that came very obvious in this data set is that typically RNA-seq, we are supposed to see a coverage of, can you see my mouse here? You can see coverage of exome very clearly here in the gene ATM. And this is what you would see, for example, from RNAs related from fibroblasts when you do a typical poly-A enrichment for RNA sequencing. But when we run this protocol with depletions, we actually had a lot of problem with a lot of intronic noise inside them. So we were working for multiple months at trying to understand why do we get in depletion from a blood sample so many intronic threads. And looking at the literature, discussing with the industrial partners, we figured out that at the end, this intronic thread seems to be aspecific. Most likely is due to the hybridization we are doing for the ribosomal depletions, and could be attenuated if we take the same library, redo a poly enrichment. But actually, the samples, they look even better if we don't touch if we don't touch at all like depletion, if we just make a poly enrichment. So currently in our workflow, we benchmark this workflow quite a lot. And we notice that despite the presence of a globin, which could be quite high in the sample, which can reach up to 80% of the transcript in the blood, we still detect at least as many genes with poly-A enrichment. And we have, for the colleague who work on splicing variant, much better results going with all blood in poly-A, integrating into the data set a lot of globin reads. One thing also here to notice is on studies that we made recently in the domain of neurodegeneration, for example, we also noticed that a lot of the genes, which are typically depleted in globin, actually they were quite interesting on the readout that we had. So working in biomarker in neurodegenerative disease, working with poly-A is definitely a big advantage here. Other developments that we were doing during the DFG calls of the project is we received FFPE DNA from very various pathology and scientists. And we knew of the challenges that some of our colleagues were working with biobank, meaning that the sample are in paraffin or they are already fixed since a long times. But we didn't really manage, despite all the tests that we make, to really find one test that tell us this is a very good DNA that we sequence, which will perform well, or this is a DNA which is very degraded and we will really have problem to sequence it. So we spent quite a lot of efforts in improving our DNA quality control and our RNA quality control, but we were not successful just measuring at fragment lengths or amount of DNA RNA to reflect the diversity we have in libraries with specific samples. This is at the moment something which is a bit problematic because we need to process large batch of sample and we know we will have dropout, but we just don't know how many will drop out. However, the bring home message here is we noticed since roughly 1 year a lot of different enzymatic strategy, especially for FFPE material, which really helped us a lot in the sensitivity. They were helping us not only for RNA samples, they were also helpful for DNA sample. And this is something I would also recommend at the moment in the workflow, testing some alternative for complicated project with degraded sample, if these enzymatic methods to repair DNA or to share DNA, if they can not really improve the success of the project. And one last point I wanted to bring also is we see with the reduction of the sequencing cost, more and more interest into whole genome FFPE sequencing for detection of somatic variants in FFPE material. Currently in Tübingen, we are setting up this workflow and we are trying to scale it up. From the clinician, there is a large interest to reach roughly 200x coverage into this sample in order to see roughly 0.5% of somatic variant in a sample. With the cost of sequencing 200x, it's still very expensive. But what we see is current workflow we have based on amplification of DNA and based on FFPE material with a relatively recent enzyme on the market. They still allow to amplify and to sequence relatively decently deep genomes. What you can see here on the figure below is that we sequence a couple of genomes which were amplified from FFPE DNA. And we could see that if we sequence them at 30, 40, 50, 60, even 70x, the number of usable reads compared to the number of base sequence is still relatively linear. So we are hopeful that with this method, we can continue scaling up deeper sequencing. And we think also that at the moment, the right strategy to don't hit a log phase where you would sequence much more, but don't get new molecule to be sequenced. Still continuing in the project of the DFG Center, one of the big challenges that we faced and did not manage yet to implement a solution to the scientists is how do we allow scientists having very different biosamples to make single cell sequencing in different centers in Germany. Before this large consortium, we had experience on how to make single cell sequencing with a researcher locally, mainly based on our research fields. But it was not very simple to allow scientists who are making experiments 100 or 200 kilometers away to come with their sample and make single sequencing in our center. What is interesting is I remember like the talk from Malte based on this paper from Kao, where they made the single cell sequencing of the mouse embryo to identify the TAD which was involved in developmental disorders. And where basically already explained us like 4 years ago, this idea of multiplexing cells and then making different indexing on the cells using pipetting that you put in specific well, add to the cDNA a specific index, repool the cells together, mix them well, reput them on a plate, and basically extend an artificial index on the cDNA, which allow after a couple of mix and pool to have a very long index, which is unique for single cells. This method at this time, when you presented it, was like totally amazing. You made experiment with 2 million cells, which is still nowadays very impressive, but 4 years ago was totally amazing. And we are also very excited to see that this kind of applications, they are coming commercially. So they are commercially available, which for us doing large scale projects is more meaningful to have better reproducible data, but also to be able to tackle other biological problems. So this is at the moment where we are scaling up, helping the user to freeze the sample at the end of the experiment, keep the sample frozen in the fridge, and at the end of their experiment, send us the samples that we can process all of their sample at once in a large batch, which reduces the cost and also makes the experiment possible wherever in Germany. From our experience, we still have a couple of pitfalls that needs to be solved, and I think our colleagues might be aware of it. Obviously, these systems of fixation, they miss some cells, so not all the cells are being detected. And this is a bit problematic for samples which are very rare cell population or very low number of cells in total. At the moment, using this method, I'm still not really convinced that if one has a sample of 1,000 cells, that one can make single cell sequencing based on this method. The second point also is this method, in my experience, are not very well designed for very different cells, meaning if a user is doing an experiment on fibroblast and on much smaller cell types, they don't mix very well, and it's not really possible to have experiment with very different cell types inside the same setup. And finally, it's tricky to handle this very large number of frozen sample in a core facility, because obviously when they are sowing, they are also being degraded. So at the moment, we're trying to optimize this workflow in order to be able to handle more than 50 samples at once. Another topic which is getting more and more popular and that we are also continuously developing is bringing RNA sequencing from short read to long reads. It was very, very nice to see that in a large population, expression of RNA has some variability, and splicing of RNA seems to be a bit less variable also in this population. So we think also that isoform expression is totally important in the explanation of genomic variants and phenotypes. And here I put an example of specific case where we had an affected index which had a premature stop codons, leading basically to, in this specific case, a reduced protein and not fully functional. And that could be validated relatively easily with long read sequencing, as you can see here, or the transcript stopping at this region. And in the unaffected, the transcript going through. So in order to make this possible, there are still some limitations. Our experience is long read sequencing of RNA based on polyenrichment works very, very well. That's a solid design for the experiment, and it works on many different kind of samples. So we had success with fibroblasts, organoids, iPS cells, PBMC. And in our hands, the cDNA amplification is a must. Like, we need to amplify the cDNA in order to have a very high quality cDNA that we can sequence and have a lot of sequencing data that can be used later on for the analysis. What we see as a challenge and we are working on is how could we make it for globin depletion or for any kind of other depletion. So this method at the moment, it don't really work well in our hands, and we are working on it since a long time now. And the second problem that we see also is that we have quite good algorithm, or we have quite good experience to predict how deep should we sequence when we search for gene expression. But at the moment, we are still struggling a bit with the tools and with the statistics in order to recommend how deep should we sequence in order to detect most of the abundant isoform. Still on long-read sequencing and on RNA, there is also from our side in our research a growing interest toward how to make long-read sequencing possible for a single-cell sequencing. One of the applications which is totally exciting is the characterization of mosaicism. For example, in a spatial individual, if we would have a trisomic conceptus which end up in a mosaicism into 3 different cells which have 2 different haplotypes, it would be very interesting to know which kind of cell type express which exact haplotypes. And this is something that can be only done in long-read sequencing. However, at the moment, we are still struggling with how much coverage is really needed to detect this. And we are seriously considering if all transcriptome is the right way to go, or if we should also work on a way to enrich transcripts that we are interested in. So this is at the moment one of the applications we are working in our research for single-cell long-read RNA sequencing. On long-read sequencing, we also had quite a lot of interest by groups who wanted to finally sequence specific locus, which are quite difficult to sequence with normal short-read sequencing. We made a study 2 years ago on sequencing the GBA gene, which has 3 other genes which are very similar and which are very difficult to detect. Typically, working on PD research, one would look at variants which are related to the phenotypes using a relatively cheap and efficient neurochip array. But this array does not really work well for this specific locus. And the group with whom we were working developed with us a PCR for this specific locus. And we can detect very reliably on it that we could have validated all the variants that we detected using this system. So Amplicon for one locus was a very efficient way to work. However, we are also more and more aware that colleagues working on specific disorders, they might want to enrich more than one locus. Typically, they are working on locus which are quite complicated, which are maybe not necessarily already existing in hybridization long read. And at the moment, we are trying to find a solution for them on how to go with an hybridization system in order to capture more than 10 genes on long read sequencing. And last but not least, as it was presented, there is growing interest into spatial transcriptomics, especially now that sequencing is getting more accessible and printing on silicium is becoming more and more efficient. We see that there are new products available which allow to make untargeted spatial transcriptomics. One of the projects we are working in our institute is we are studying a disorder of the brain development. And we are particularly interested in the development of the striatum in this animal. So what we made in these systems is we used one of these systems which allow us to hybridize transcripts to flow cells in order to amplify on a flow cell transcript capture from the tissue. What you have here is the representation of the brain that we were sequencing. Because this method has a very high resolution, it has 220 nanometer between spots and 500 nanometer from center to center. One can detect roughly 400 spots per cell, so single cell resolution is not an issue on these systems. Usually, what we do is we rather do an analysis of the single cell system based on nuclei, or we do a bin of 50 of these hybridization spots. And what we can nicely see on it is that the system gives different neuronal types that we can identify, which are also histologically meaningful, and that we can enrich in order to detect what a specific mutation in the brain leads to the striatum development of rats. So that's the end of the presentation I want to bring. It was the work of a lot of people inside our sequencing center. It was also a lot of collaboration within the consortium to have this method tested and implemented, and I thank you for your attention.
Well, thank you, Nicolas, for going through the world of the centers. That's one example. And I guess the other ones are in a similar situation at the moment, and they have a lot of exchange to see where the whole network is. Are there questions out there in the web application? No? Peter.
I saw a shift to epigenetics also from one year to the next. Is that a trend in general, or what's your opinion?
So I think in general, epigenetic is a very dynamic field where there is a lot of requests inside it. However, we also see that epigenetic methods are so focused and they are so complex to have running that generally a few very high profile groups are very expert inside it, and we really have problem to re-implement this system by us. First, because there is not enough requests that it's really meaningful to re-implement it everywhere. And second, because technically one need to have very specific workflow running, including extremely specific bioinformatics. The need in epigenetics is there. We see a lot of scientists who would love to make it. I still think that they are blocked because of difficult bioinformatics, difficult experimental setup, and not many researchers can really do service for them for it.
I have the same feeling. Excellent answer. Nicolas, I don't see any more questions. Thanks you again. And we come to the last talk of this session for Alexander Vogel from Oxford Nanopore. As I said, that's the tradition of the meeting. Every session has one talk from industry at least. And now we go to hear about the recent advances in long-read sequencing with nanopore.
Yes, good afternoon, everyone. My name is Alexander. I'm a senior field application scientist with Oxford Nanopore. And essentially, I'm the person keeping you from your well-deserved coffee break. But I will try to make your wait worthwhile. As Nicolas already said, Oxford Nanopore is known as a very innovative company, so I could easily fill an hour talking about cell-free DNA, talking about direct RNA, talking about single-cell sequencing, but I only have 15 minutes, so in preparation of this event today, I had a look at the agenda, and I was very positively surprised to see an agenda, but also an event that embraces change as this one does. First of all, you're trying to influence the decision-making via the Modellvorhaben. We have a great lineup of talks with many people using long-read sequencing, not only within Germany, but also in Denmark, for example. And my personal favorite, you're giving the poster prizes in Bitcoin, which I really liked. I'm not sure if the organization committee is aware, but the Bitcoin price since then went from 24,000 euros per Bitcoin to 32,000 this morning. So I'm just leaving that there. I, from a technical end, have a similar experience when I talk to customers, to users of Oxford Nanopore at at events, but also at conferences where they ask us, oh, you can really do this nowadays. So what I would like to do today is I would like to give you a little bit of a snapshot of what is happening, what are the latest advancements in terms of nanopore sequencing, and how this may apply to the work that you're all doing. For the ones who are not yet convinced that things need to change, let me pick you up. First of all, there are more than 1/3 of the disease-causing or the known disease-causing variants that are caused by not only SNPs, but by variation that goes beyond a single nucleotide polymorphism. On top of that, these are 30 times more likely to impact the gene expression, so have a more sophisticated impact on the downstream effects. As we all know, structure variation tends to be much, much more complex than just single nucleotide polymorphisms. They can range within a couple of hundred bases to, as stated here, more than 5 megabases, where long reads come in particularly useful to resolve these and to accurately pinpoint the breakpoints of these large structural rearrangements. On top of that, we have not only structure variation, but also single nucleotide polymorphisms hidden in what's referred to the dark region of the genomes. This could be regions that are not accessible by short read sequencing due to their repetitiveness of the genomic contacts, or maybe there's a PCR bias that is used either in library preparation or in sequencing as well. And then last but not least, if we're looking at the aforementioned epigenetic space, then there are solutions for this as well, but the common arrays only cover less than 3% of the sites, so there's a lot more variation to explore. We of course have solutions for this. So if a patient comes to you and asks for a diagnosis, of course there's a first clinical assessment, and then based on a hypothesis, there are various different tools out there that we can use to look at repeat disorders. This could be flanking PCR, this could be Southern blot. There are other methods available if we want to look at copy number variation. If some of these, or sometimes the hypothesis isn't confirmed, so in these cases, there's of course the option to do a clinical exome. If they still come back negative, there's the option to do a short read whole genome sequencing. But of course, this all costs a lot of time, a lot of effort, and in the end, only 40% of these cases actually get a definite answer. Okay, this is a variant that we know is associated with a certain disease. So we think that there's obviously a need to change, and we think that we as Oxford Nanopore can contribute to this. This is just an example from a time perspective, but I will also walk you through some other benefits of the technology in the next slides as well. So of course, this journey, this diagnostic journey can sometimes take years really for the patients until they have an answer. There are some settings where years just don't cut it. There are settings where we need to have a diagnosis within hours, especially if we're looking at the last one, which was done at the University of Washington in Seattle, when it is about newborn genetic screening, where there are newborns on the ICU, and we need to know as soon as possible, okay, what is the cause of the condition? And there the team has shown that with nanopore sequencing, they could do this within 3 hours, which can really have a clinical meaningful impact. Taking one step back, what is so different about our technology that we can think we can help you to contribute to this change? First of all, we are directly sequencing native DNA or RNA, meaning we don't need a PCR in the library preparation process, but we also don't need a PCR in the sequencing process. What we do instead is we have our protein or motor protein, which feeds the double-stranded DNA molecule through the nanopore. At the same time, we are applying a voltage, and we are depicting, or we are measuring changes in this ionic current flow, and this is what our raw signal is based on. So sequencing native RNA, native DNA, no PCR amplification, which means that we are essentially read length agnostic. So we can sequence very short fragments, 150 base pairs, cell-free DNA, no problem, on the same device as we can do 100 megabase, 1 megabase, 5 megabase, 4 megabase, 20 KB reads. It's completely up to you, or to be more precise, to the application or the scientific question that you want to answer. The fact that it's PCR-free also enables us to depict base modifications, not only 5-methyl-C, but also 5-HMC, 6-methyl-A, and that not only in CpG context, but we have also all context model where we can essentially look for all of these variants or for all of these methylation patterns across the whole genome and not only in the 3% of the genome that common error is referred to. Real-time analysis, we have seen that time can be the essence of an analysis. So there we can analyze the data as it is generated. So while the sequencing run is still ongoing, we can already start to analyze the data and to derive meaning from it. We have a great example from this presented, I think it's tomorrow, by Helena Kretschmer, who have really taken this to an extreme, and they can do brain cancer classification within less than an hour. So previously we've seen this 3 hours. So we have one group presenting later where they've even cut this to below 1 hour. The nice thing about it is that the system is incredibly scalable. What does this mean? We have, of course, a different range of devices. We have 3 different kinds of flow cell: fungal, MinION flow cell, and PromethION. While fungal and MinION flow cells are compatible with MinION and GridION, we have PromethION flow cells. Those are generally ones that are used for whole genome human sequencing, where we then have also a lineup of P2. This is a device that can sequence up to 2 flow cells in parallel. Its bigger brother, the PromethION 24, with 24 of these positions in parallel. And then the production scale sequencing device P48, which can analyze up to 48 of these flow cells in parallel. What can we actually do with this data, or where can we put this to use? One of the applications of many is, of course, repeat expansions. Repeat expansions are commonly referred to of 3 nucleotide repeats, and then we can associate them to a certain disease where we know for a healthy individual, we expect this amount of copies of this repeat. And then if we have a larger copy or a diverging number of copies, we can associate this to a certain disease. So far, so good. Why is nanopore sequencing particularly well suited for this? Of course, because of the long reads, we can cover these repeat patterns, and we can not only say, okay, is it binary? So is the patient impacted by a repeat expansion? But we can also accurately determine how many repeats are present for this particular loci. And on top of it, we can also start to look for variation in these repeat patterns. We have now seen that it's not only the repeat number or the number of the repeats, but also there can be hidden variation that maybe one nucleotide in such a 3 nucleotide is changed. And we can now depict exactly what is the position of this variant, and does this have, or does this alter the prognosis for this particular individual. Just as one example, we have here the Fragile X syndrome. You can see on the right, we have, or in the IGV screenshot, we have 1 haplotype. So in this case, we are taking additional advantage of the long reads to phase the 2 haplotypes out. So on the top, we have a short repeat version, which is considered the healthy or the unaffected allele. And then on the bottom, you see the second haplotype, which carries the long repeat pattern. However, surprisingly, this patient still exhibited the symptoms of the Fragile X syndrome. And what the team then did is they looked beyond the repeat pattern, and they also took advantage of the base modifications that we detected, or that the system can detect at the same time without the need to change anything in the library prep, but purely bioinformatically. If we do that, and if we overlay this with the exact same loci before, we can see, although that we have 1 healthy allele, we can see that both of them are methylated. So in essence, the FMR1 gene is not expressed based on the methylation pattern that we see here. We also know that there are loci, depending on their methylation patterns, they can contribute to different diseases. I've already seen a poster by Matthias Linke and his group that is talking about much more about the Angelman syndrome. So I don't want to go into too much detail here, but we can see the same loci, whether it's methylated or unmethylated, can entirely change the prognosis or entirely change the disease that is caused by this loci. Same, of course, is true for structure variation. If we look at structure variation, there are small insertions and deletions that need to be detected, but sometimes they can be really nested complex structural rearrangements that can span quite a significant amount of bases and are thus very, very challenging to resolve with short read sequencing. There, of course, the long reads help for A, placing the reads more uniquely in the genome, and B, to be able to reassemble these complex regions and to get a better picture of what's actually going on in these particular regions. In contrast to the CMAs that are generally used, we also can pinpoint the breakpoints precisely. So we are not depending on a given interval by the probes that we have on such an array, but we get the information across the whole genome and can pinpoint the breakpoints precisely. Interestingly, the example that's shown here on the right is not actually from a PromethION sequencing run, but that was a sample or the mother and the proband were actually sequenced on a single MinION flow cell. So you can see that even works with very low sequencing depths where you can accurately depict such a large structural rearrangement. We have already seen a little bit about the benefits of the phasing in context of the methylation, in context of the repeat expansion. But of course, this is also a powerful tool to look at SNPs or to look at the context of different variant types. Here we have the example of two variants that we have in the same individual in the two different haplotypes. Once they are split out in the haplotypes, you can quite clearly see that one of them carries a structure variation and the other one carries a SNP. But without the phasing information, of course, sometimes it's difficult to know, okay, are both of the variants in the same haplotype? Are they in different haplotypes? So there as well, the long reads help to put these variants into context, into the right phase to see, okay, is this actually one allele or are both alleles affected by this given variant? We can do copy number variation crawling as well. We can do this on a quite broad range. So we can do very fast aneuploidy identification within less than 3 hours. We can also resolve smaller copy number variants like less than 500 KB, or of course, if we sequence more deeply toward 30x, we can also pinpoint the breakpoints of these copy number variants and can look into interesting aspects, as mentioned before, mosaic copy number variants, for instance, and there are also indications that these are associated with known diseases. Just as a brief summary in between, so why do we think that nanopore is worthwhile looking at? One platform gives you all the read lengths, gives you many answers to a lot of the different variation that we see within the human genome. Looking beyond the SNPs, looking beyond indels, covering complex structural rearrangements, copying copy number variants, looking at repeat expansions, looking at mosaicisms. We can also look at mitochondrial, and of course, we have the haplotype phase on top of it and the methylation on top of it available, which I think is really a comprehensive offering to look across the whole genome. To make this as straightforward as possible, we have prep solutions available. We have, of course, they are compatible with all our sequencing devices, so no matter if you're running it on a MinION or PromethION, the prep will be the same. And then we have data analysis options that are plug and play compatible with our WF human variation workflow, which lets you analyze all the what I mentioned before: SNPs, structure variation, methylation, repeat expansions, copy number variants, all within one workflow. The execution of one command essentially provides you with all of this information to then analyze further and put in context with healthy individuals or a database. Just two last things. One, scalability. I've mentioned this already, especially with the PromethION. The P24 does have 24 positions, so it allows you to sequence roughly 2,500 samples per year or flow cells per year. For the P48, logically, it's twice the number of that. For the P2, it's roughly 200 per year. So from a P2, there's the potential to upscale to a P24. If you need less sequencing depth, there's also the option to multiplex 2 or 3 samples into an individual, into one flow cell, which of course increases the amount of samples that you can analyze within a year. So in terms of scalability, I think no matter what scale you're looking for, there should be a solution in the lineup or in the setup that we support. Last but not least, I promised the latest and greatest benchmarking results just to show you where we are currently at. Simplex accuracy at 99.5%. This has been largely driven by updates in chemistry, but updates in the base calling model as well. We are measuring now at 5 kilohertz instead of 4 kilohertz before. We have a new feature which we call duplex, where we are taking advantage of reading both strands of the same DNA molecule, and like this come up with reads that have a significantly higher raw weight, or not raw weight accuracy at this point, but accuracy of more than Q30 at this point. For assembly purposes, the long reads, the accurate long reads help, of course, to get a good consensus accuracy, but also if we're looking at all the variant types, maybe SNP in the structure variant or methylation, we think the F1 scores speak for themselves, that we are on par, if not better, for many of these with short bit sequencing. I personally like IGV screenshots much more than I like numbers. So just to put a little bit of more visuals to the numbers that I've shown before. So those of you who are familiar with our chemistry for quite a while, this is what we call the older chemistry, the R9.4.1 flow cell with the Kit 10 chemistry. And every dot that you can see here represents some sort of an error or variation. And there you can see the clear improvement switching from this to the latest Kit 14 chemistry. And then if we are looking at the duplex reads of this chemistry, we can essentially see almost perfect reads with a Q-score of 30. And with this, I come to an end. I hope I could show you where Nanopore currently is, how we hope to contribute to making this change in diagnostics or in the field of human genetics as well. And I'm happy to take any questions. Thank you.
Thank you very much for this concise overview and the news. Are there questions from the audience? Stefan, from the web, is there also something? No. Okay, Stefan.
Yeah, great. Can you tell us a bit more about the duplex reads, the future of the yield, and how they could be further improve the yield, maybe?
Yeah, definitely. So it's very actively being researched currently internally to increase the rate of the reads that we turn into these duplex reads. So at the moment, what you can expect from a flow cell is somewhere between 20 and 40% of the reads that are generated can be turned into like a duplex read. What we have in the works at the moment and what we have in a closed developer program at the moment is what we call high duplex flow cells. So these are 10.4 flow cells, which are specifically optimized to increase the rate of duplex reads. And this is one way of increasing this. The goal here is to go to up to 80% at some point of the data that is generated per flow cells to turn into this duplex read. However, for most of the applications or for the variant types discussed, we see that the simplex reads do a very good job already. Where the duplex reads help is, of course, if we're talking about genome assembly or if we're talking about indel variants, for instance. Yes.
Can I ask a short question? The $345 euro genome, how long does the sequencing take?
The sequencing by default takes 72 hours. So that's the default runtime that we have. But I said, thanks to the real-time sequencing, you could also think about stopping a flow cell or stopping a sequencing run after you have acquired 90 gigs of data, 30 gigs of sequencing depths, or once once you have the answer to your research question that you started with. Yeah.
Super, thank you. I guess everybody wants to have coffee. We have a nice coffee break until 4, so plenty of time, and he's also here, so if you have more questions.
Indeed.
Thanks to all speakers. That was a great session.